associated with aging.
Hello, good afternoon everybody. So today the topic of my talk will be primarily on aging. So age is the primary risk factor of basically all chronic diseases. If we look at the incidence of cancer, of respiratory diseases, myocardial infarction, stroke, dementia, diabetes, they all increase exponentially with age. So this means that age is the strongest common risk factors for all the major chronic diseases that are the major component of mortality. And there is no other single risk factor, you name it, BMI, blood pressure, glucose, a genetic predisposition that comes not even close to the effect of age on diseases. And this means that if we would be able to find a treatment that slows down the ticking of biological age, we would have with one single preventive treatment a right shift of all these curves. And this is really a Copernican revolution in the field of medicine, because it means that shifting from the current medicine approach, which addresses the terminal molecular pathways that are activated during the disease, from intercepting the single common risk factor of all those diseases. Now, the good news is that it is indeed possible to influence aging by modifiable factors. And the most spectacular example are the studies of calorie restriction in primates that have shown that restricting calories delays significantly and quite strongly the onset of age-associated diseases in a primate. However, as you see, this experiment took 3 decades. So the normal way to get around this problem is to use like worms, nematode worms. They're very convenient, but I don't think to convince you that we are very different from worms. So if you have to use vertebrates like mice, zebrafish, and even worse, dogs, monkeys, those experiments take up decades, and this is a biological limit that is simply impossible to overcome, no matter what your technology is. Even worse, the situation is in humans, because we cannot think about starting now a treatment, let's say with metformin, and see in 30 years how many of the people who took metformin for 30 years are still alive. So it is necessary to find some biomarkers that are predictive of effects on the long term and that allow to perform experiments in a limited time frame. And this is the concept of the aging biomarkers. And there are actually plenty of aging biomarkers. So here you see a number of different physiological parameters, from BMI to maximum oxygen consumption, to maximum lung capacity, cholesterol, triglyceride, lipoprotein A, renal function, blood urea, this is glycated hemoglobin, leptin. They all change gradually with age. However, each of these biomarkers only captures one specific aspect of physiology. And assessing all those biomarkers at the same time is not a scalable approach. And this is why many different groups have used omic technologies, so RNA methylation, proteomics, or transcriptomics, in order to derive so-called molecular aging clocks. So what are molecular aging clocks? Are machine learning algorithms that are trained to predict age based, for example, on patterns of DNA methylation. And a very simple assumption under these models is that if your predicted age is higher than your real age, then you are at risk of disease or you are already sick. Whereas if your predicted age is lower than your real age, then you are in a particularly good condition. And I mean, what people are looking for are interventions that reduce the biological age. I mean, we know for certain the number of diseases will increase the epigenetic age, and now, I mean, these biomarkers are used in order to find interventions that reduced the predicted biological age. So we have developed a three-pronged approach to this problem, which is based on an innovative animal model, on a proprietary complex AI architecture, and what we call a hybrid strategy for the identification of anti-aging drugs. So the animal model that we use since 20 years is called a killifish. It's a fish. That means it has bones, kidneys, heart, you know, all the typical bodily structure of a vertebrate, but the lifespan is more similar to those of a short-lived vertebrate. And the work from my lab and several other labs have shown that these animals display all the typical hallmarks of aging. I'm here showing only a few. This is cardiac hypertrophy. This is the heart of a young fish. This is the heart of an old fish. Cellular senescence. This is a staining for SA beta gal, the marker of cellular senescence. And spontaneous neurodegeneration. The red dots, if you can see them, these are accumulation of protein aggregates in neurons of the locus ceruleus. So these are the first neurons that are hit in Parkinson's disease in humans. We and others have shown that you can use this model to test effects of drugs. For example, resveratrol was tested in this model and other drugs. And the genome is sequenced, and it's possible to do transgenic and genome editing with CRISPR-Cas9. So it's a full, complete model organism. What we have very recently developed is also a protocol of ex vivo brain aging. So we can dissect the brain of this fish and grow it in culture for about 4 weeks. Now, 4 weeks in the killifish, if you scale it up, it's decades in humans. And here you can see, for example, that treatment of these ex vivo brain cultures with beta-glucans causes a very strong increase in the number of mitochondria. So this is an ex vivo model that can be used for drug testing, and in very limited time frame. But the problem of the epigenetic clocks that are the most used clocks is that they are not transferable from one species to another. So they're very precise, but they're species specific. So we developed epigenetic clocks for the killifish. Here you see the prediction of age in the test set. And there is an error, so the maximum life, the median lifespan is 40 weeks, and there is a median error of less than 2 weeks. But more strikingly, what we can measure is not, what we can predict is not only age, but really health condition. So we did this experiment where we took fin samples of fish at 2 different ages in young life, and then we knew the day of death of each of these animals. And then we tried to predict the exact age of death. And this is the prediction of the age of death. So we can honestly, with astonishing for us precision, identify that the health status at early age, which then is reflected in the mortality risk. However, if we want to transfer this from fish to human, we have to resort to RNA. And RNA is much, much more difficult a beast to tame than DNA methylation. So to do this, we developed a proprietary architecture for a neural network that uses the same genes to predict age in fish and in humans, and has a very important characteristic that we can use data from different tissues with just a single model. And this is just an example. In blue is the training set, in orange is the test set. These are 6 different tissues. It's one single model with 6 different tissues. There are a few outliers, but all in all, it's a very precise prediction. The error is 2.2 years, that is only slightly higher than the error that we have with epigenetic clocks. So, what is the competitive advantage of a transcriptomic clock? That it provides a direct readout of gene expression. So I know which genes are responsible for the deviation from the predicted age, and I can reconstruct the function that is related to this gene. The other point is that it can leverage on huge public data sets of interventions in humans, whereas the same data sets are not available for epigenetic data. It is usable basically not only in human, in fish, in mouse, in horse, in cat, in dog, you name it, across all vertebrates because of the core set of conserved gene, and has a high precision in predicting lifespan. So, what we propose then, it's a method by which we either do the ex vivo aging approach, or we take living animals, and then before starting the treatment, we take one piece of the fin, then there is a drug treatment for 3, 4, 5 weeks, and then we take the second piece of fin, so we have 2 prediction of age for the same animals which are spaced a few weeks apart. Now, what you expect in control condition is that, of course, the age increases, and I mean, if it's 5 weeks of 5 weeks, and in the treated animal, if the drug has the desired effect, you expect a reduced increase of the predicted age. On the other hand, if we do a prediction of time to death, this will decrease with time in the controls and less so in the treated. So this is a platform that we now have developed and that we hope to use in partnership with companies that have interesting compounds or nutraceutical formulations in order to identify putative anti-aging compounds, so compounds promoting health in old age. But then we can also use the same system for discovery of new compounds. And this is because we can use our transcriptomic clock to predict the transcriptomic age of human cells. And there are huge data sets on the effects of compounds on human cells at the transcriptome level. So we can identify compounds that reduce the transcriptomic age in vitro, and then use this compound in vivo, do a first test on the predicted age, and then take the lead compounds to really measure effects on lifespan and a series of aging-related markers. So, again, a few words about the traction of this model. Probably no one of you has ever heard about it, but it made it into the News and Views of Nature, and this is the number of papers on killifish as a function of the year. These are the number of labs in the world that are using killifish for different aspects of biomedical research. This is our team, so it's me, then Daria, who is the commercial lead, MBA, and the person who hopefully will be in charge then of developing this startup, and then Debdash Paul is the AI person, and Luca Dolfi is the animal technology person. And I finish by thanking all the people of my lab, which are, some are here in Italy, some are in Germany, and if you have any question, this is my contact, and this is the contact of lachesis.bio. Thank you for your attention.